This is going to be an overview of gene technology. So, the main aim of the new development of gene technology is to determine all the different genes that are contained in your genome. So a genome is all the different genes that you contain. So it would be the gene for insulin, gene for glucagon, basically all the different genes that you would find in a human. The proteome, on the other hand, is what those gene products are. Now, sometimes your genes don't code for proteins, sometimes genes actually code for tRNA, or maybe their rRNA. So it's really difficult to determine the proteome from human DNA because of all the extra bits of DNA that we have that code for other things that are not actually coding for proteins. But proteome is all the different proteins, so that would be insulin, glucagon, hemoglobin, etc. Okay, so once you've identified your genome and you know what all the sequences of bases code for, you can then use that. So if you, for example, let's take insulin, that's the simplest example. So if we want to make lots of insulin, so originally insulin was taken from animals. The problem with that is the insulin from a pig or a cow is just not the same as the insulin from a human. And that's because when you look at the receptors and the shape of insulin in humans versus pig, it's slightly different. And so therefore this one in a pig would work perfectly, but if you put it into a human, then that insulin wouldn't bind as effectively to the receptor, and so you might need a higher dose than normal, and so that's not as good. So pig insulin isn't 100% complementary to the receptor found in humans. So that's a problem. But what you can do is you could take either the gene sequence, so now we know what the gene is for insulin, we could use the DNA and we could chop it up, so chop out the gene, PCR it, so make lots and lots of copies of it, and then insert it into a vector and into a bacterium and make a transgenic bacterium. Now, issues there are if you start chopping out bits of DNA, you might get mistakes in there. When you amplify it up with PCR, there's more mistakes. So a better way would be to take the beta cells from the pancreas, because they will have lots of mRNA, which we know are a true copy of the DNA. They also don't contain, if it's mRNA, any introns. So when you multiply it up with PCR, you've got no issues there. But before you do that, you have to go through and make cDNA. So you take your single-stranded mRNA and you make it into cDNA, then you amplify it up, insert it into a vector, put it into your bacterium, and then you've made your transgenic. Okay, the last way that you can make your gene that you want to put into your bacterium is using a gene machine. Now, a gene machine is a relatively new tool. All that you're doing there is you know the sequence from your genome, so you've got your genome, and you take the gene for insulin sequence, so whatever that is, whatever it is, make it up. Okay, and then you put that into your gene machine. You supply that machine with nucleotides, and it basically figures out how to make sure by making little overlapping strands called oligonucleotides, to end up making a piece of double-stranded DNA with no introns. And that's because what you did to get this is effectively you can get your code from, so we start with this is our insulin protein. Then you sequence the amino acids. Then you work out the mRNA based on what the most common codons in humans are for each amino acid. So, for example, leucine has 3 different codes, AAA, AG, and AAC. And in humans, the most common form is AAA, say, for example. So that means we know that if we see a leucine in the protein, then it's probably because it was coded for by AAA. So you make up your code, you put that into your gene machine. Because it's made straight from the protein, you're going to end up with no introns. The gene machine uses its nucleotides, which it makes into oligonucleotides, which are just short fragments. Now, why is it not using single strands, single nucleotides and putting them along? Basically, it's just because it's more likely to make mistakes. If you have little groups, if you start putting together more than, say, 10 or 20 bases together, you start to get mistakes incorporated, so basically changes in the base sequence. So it's better to have shorter fragments and then try and stick them together with overlaps, and that will help to reduce the number of errors you incorporate.